guys, welcome back to our channel Explore Biology. So we have been exploring different concepts in our channel. So if you have not watched the previous videos, please do watch it. I have done a wonderful videos on developmental biology. Please have a look. Right. So from this videos, I'll be starting a new chapter that is body fluids and circulations. Yes, in few videos, I have added some of the topics of this chapter, but now we are going to start from the basic part. Let's see. Let's start the session now. So don't forget to watch the video till the end to understand the concept very well. Right. Now, what is this body fluids? See, the term itself explains that it's a body which contains what? A fluid. Now, why this fluid is that much important? That brings a question, right? Why do you think body require these fluids? Why is that important that we study? See, when you understand that we are complex organisms, if you take amoeba, it has a single cell and there is not much difficulty in transporting nutrients to them, right? But consider us. We are huge personalities with trillions of cells. Now, in such case, the nutrients have to reach each and every cell. Now, that is difficult. So there has to be someone like a train or a aeroplane which is carrying things from one place to another. So inside our body, these fluids does that. So body fluids help in circulation. Circulation of what? Circulation of nutrients, ions, electrolytes, many different things through them. So in our body, we have two different types of fluids. So that we are going to study today. Let's start the session, children. Right. So what is the fluid which is present inside our body? That is nothing but blood. So blood is the fluid connective tissue. So in animals, we have four different types of tissues in which one of the tissue is the fluid connective tissue. Okay. One of the tissue is connective tissue. Inside connective tissue, we have many different things. Slowly, we will be studying that also. Don't worry. Right. So does this fluid do? They help in transport. So they act as transport system in higher animals. Remember that. In lower non-chordates, you don't see much circulation like till ASCII elements. Okay. From Annelida, you will see the circulation, right? The circulatory system, but not that developed one, right? So this fluid that is blood, it is not simply made. It contains two different components. What are those components? One is the majority one that is plasma. So plasma accounts about 55% of the blood and we have cell components which is of 45%, little bit less. So much of competition between these two. So majorly what is present inside the blood? Plasma. Now, what does this plasma do? Don't worry, slowly we will be learning about it, right? So the plasma is a fluid. The cell components are having different types of cells. So it's not like there is only one type of cell. There are different types of cells, majorly categorized into three. That is red blood cell, white blood cell, and platelets. So one by one, we will be studying about them also. Let's move on. I hope this you understood. Chal. The first component in the blood, that is plasma, right? Very, very important. How much percent it accounts? 55% of the blood, right? So this plasma is a straw-colored liquid. Straw-colored liquid, you can see in the image given here, you can see three different layers, right? In that, the straw-colored layer is majorly present. Then we have a buffy coat. Then we have a red color layer, right? So this straw-colored layer is nothing but plasma, right? So plasma, if you see the pH, very, very important, children. What is the pH of the plasma? So the pH of the plasma is 7.35 to 7.45, which is almost neutral, right? Not much acidic, not much basic. Neutral component. So what does this plasma contain? It contains water, electrolytes, ions, clotting factors, hormones, vitamins, etc. Many different things, in which majority is water, right? Majority of it is water. Now, if you remove clotting factors from that plasma, that will become what? A serum. So a serum is what? A serum is equal to plasma minus clotting factors. Simple. Plasma minus clotting factors. So if you remove the clotting factors from plasma, that becomes a serum, right? So the upper layer we find is the fluid, which is made of plasma, right? Now, what is the middle layer? It's buffy coat. There is nothing colorful present there. So it's simply the WBC and the platelets, the cell components. And at the bottom, we have red color one. We all know what is that red color. It's RBC, that is erythrocytes, right? Let's understand one by one about these cells. Before going into that, let's understand what are the uses of these components which are present in the plasma. Why is it required inside the blood? Let's see. So the functions of plasma, majorly you have to remember, is the clotting factors. Very, very important, children, for coagulation. For suppose on a very fine day, it's a holiday for you and you wanted to help your mom and you ran into the kitchen and you started cutting the vegetables. Suddenly the knife got cut, your finger got cut. So what happens? Blood oozes out. So what you will do? First, you will cry. Mommy, I got hurt. So what happens then? Is that blood coming out continuously? No, it won't come continuously. Why? Because inside the blood, we have these heroes. Who are those? Clotting factors, which will be helping in what? Which will be helping in coagulation or clotting, right? So what they do? They immediately go there and block that and immediately stop the flow of blood, right? So they are literally heroes, right? The second important function is the presence of albumin. So inside the blood, the osmotic balance has to be maintained, which directly helps in maintaining the blood pressure, blood volume, and all. So for that, who is required? Albumin is required, right? This is the second main function. And we all know blood is acting as a transport system. So it carries food materials, that is nutrients, throughout the body. Right? Very important functions, children. Remember these things. Right? Okay. So that was about the story of the majority part inside the blood, that is the plasma. Now, what about the least part? Not that least, a little bit less part, that is cell components. Let us understand about them. So, right. The first and foremost cell, which we all familiar with, is the RBCs. RBC is red blood cell, or we also call it as erythrocytes. Okay, they are formed in red bone marrow. They are circular in shape, discoidal, non-nucleated in mammals. Usually, the mother cell of RBC will have nucleus. Later on, it get disintegrated, and now RBC won't have nucleus. You can ask me, ma'am, what nucleus has done? Why it has got removed? Because RBC's function is what? Carrying oxygen. So if there is more space for nucleus, there will be less space for transporting oxygen, right? So our body is way more clever than us. So what it has done, which is not required, it removed it, right? So nucleus is not present inside the RBCs. Only we have hemoglobin in them, a protein which helps in carrying oxygen, right? Or carbon dioxide, right? For the transportation purpose. So a healthy individual should have 5 to 5.55 millions of RBCs. And what is the lifespan? It is 120 days. After that, it gets disintegrated in the spleen, right? And the healthy person should have a hemoglobin of 12 to 16 grams, right? So these are some points which you have to remember for the multiple choice part. Okay. So circular, discoidal, non-nucleated, these all helps in the function of RBC. Based on the function, only the structure is being designed, right? So it's very, very important. The lifespan of it, what are the healthy individual, how much it should have, all these parts are very, very, okay, right. Coming to the second type of cell. What is the second type of cell? We have is leukocytes or white blood cells. See, the name itself is giving its. See, in RBCs, we have hemoglobin, which is giving the red color. But in WBCs, we don't have that red color hemoglobin, right? So that's why it is white blood cell. So it is colorless, irregular, or spherical in shape. Again, leukocytes are of many different types. Okay, that also will be studied as well. So how much it has to be? 6,000 to 8,000 per cubic millimeter. What are the two main categories? So presence of granules inside the cytoplasm divide this leukocytes into two types. What are those? Granulocytes if the granules are present, agranulocytes if the granules are absent. Okay. Granulocytes if granules are present, agranulocytes if the granules are absent. Let's see what are those. You can see here, we have the divisions. We have all these things clearly mentioned here. So the granulocytes part are neutrophils, eosinophils, and basophils. These are what? These are granulocyte, because if you see them in microscope in their cytoplasm, we see granules present in them, right? And here we have a granulocyte because there is no granules present in their cytoplasm, right? Granulocytes and agranulocytes. See, the first part is neutrophils, which is having 2 to 5 lobed nucleus. Okay, you see the nucleus is arranged somewhat like this. Okay, so this is 2 to 5 lobed. How much percentage is 60 to 65? Majority of granulocyte is neutrophil. What is the function? Phagocytic cell. What is phagocytosis, children? Eating it, breaking it into different parts. So when a foreign body is entering into our body, foreign particle, so our body has to recognize it. Hey, this, you are not part of us. You have to get out. So body will send them out. How? Through phagocytosis. So it will take up them, break it into pieces, and shows that to the lymphocytes, and lymphocytes kill. Okay. Now, the second one is eosinophil, which is bilobed. Okay. How much percent? 2 to 3. Very little. So what does this base eosinophil do? Resist infections and are also associated with allergic reactions. So you started to help your mother. So you planned, okay, I will clean my house today. So you took the broom and started cleaning. Suddenly you started sneezing continuously. What is that? Allergic reaction. So who is acting there? Eosinophil is acting. Right? And the third one is basophil, which is very, very, very less. The nucleus is S-shaped, secretes histamine, serotonin, heparin, and all are involved in what? Inflammatory reaction. So mostly granulocytes are part of inflammatory reactions, right? Indirectly, we can see that. What about agranulocytes? The first one is lymphocyte. We have two types, T lymphocyte and B lymphocyte. They play very important role in immune system. They are spherical in shape, 20 to 25% stage is their presence, and they are responsible for immune responses, right? And monocytes, it is kidney shaped. They will definitely ask this question. The smallest agranulocyte, which is having kidney shaped nucleus, is what? Monocyte. They are phagocytic cells which destroy the foreign organisms which is entering into our body. Mostly, if you see the WBCs, they play major role in protecting us. They are like our army, right? Okay. These things you have to remember. So have a look through the table. That is very, very important. And finally, we have the third type of cell. What is that? Platelets. Platelets are actually not called as a sites because see, we say red blood cells or erythrocytes, white blood cells or leukocytes. Site is a term used for a cell, but we are not saying platelet site because platelets are the fragments of a cell, right? So they are irregular and they don't have nucleus in them. There is very, very important that it has to be 150 to 350, and they are fragments of megakaryocyte. Like I said, see, karyocyte. Site is a cell, but this cell is undergone fragmentation. So these are what? These are fragments, not a complete cell. So that's why we are not using it as a site. Okay. So platelets, what is the function of these platelets? Just now I told what is the function of the platelets. They help in clotting. So platelets, see, platelets are very, very important. Whenever there is a wound, they come, they block that wound so that the blood doesn't go out. That's why a person whose platelets are falling down, there is high chances of death, high chances of bleeding, right? So it's very important that we protect that person. Okay. So that's about the blood and its components. Okay. I hope you have understood this part, right? So we have studied about few things like blood and its components. So I'll just give you a flow chart. Blood. In blood, we have two components, like I said, plasma. And we have what? We have cells. Again, in cells, we have different types, right? We have RBC, we have platelets, we have WBC, right? In WBC, again, we have categories, granulocyte, and we have agranulocyte, right? Granulocyte contains neutrophils, eosinophils, basophils. Agranulocytes contain lymphocytes and monocytes, right? So this was about the blood. Now, if I say, does this blood will be same in all the organisms? Yes, that is true. Because if I cut my hand, I get a red blood. If you cut your hand, you get a red blood. But why there is a change in different blood groups? That's important. See, we say mine is A blood group. You will say yours is B blood group. Someone will say mine is O blood group. Oh, O is very rare, we say. Now, what are these blood groups? So, in our body, every cell has a recognition marker. So, I said that if a foreign particle enters into our body, now how our body knows, oh, this is foreign particle and this is our body part. How does that know? It knows that. It depends on the antigens. So in our body, on the surface of the cell, we have antigens. So for example, this is a cell. This cell contains some markers on it. These markers are nothing but antigens, right? These are antigens. Now, in RBC, not in RBC, on the surface of RBCs, they have found three different types of antigens actually. First, they have found two. Then later on, they found one more antigen. Okay. So the two different types of antigen were found, that is A antigen and B antigen. So based on these antigens, we have four different types of blood groups. How? Let us see. See, first suppose if the person is having A blood group, okay? If he is having A blood group, he has antigens A. That means on the surface, he will have A antigen and the body will synthesize an antibody against that antigen. So antibody will be B, but antigen will be A. Okay. If it is B blood group, antigen will be B, antibody will be A. Reverse condition. A blood group means A antigen. B blood group means B antigen. If antigen is A, antibody will definitely be B. Then what about AB? So AB blood group means that means it should have both A and also B in them. Then what about antibody? Now body gets confused. Are A antigen is also there, B also there. Now what antibody have to synthesize? So it will not at all synthesize any antibody. So antibody is absent in AB blood group. Now coming to O blood. O blood group will not have any antigens. See, on the surface, there is no antigens. So what body will do? It will synthesize both the antibodies. Okay, there is no antigen, I'll synthesize both antibodies. So it synthesizes both A and also B, right? So A blood group will have A antigen, B antibody. B blood group will have B antigen, B antibody, right? So here you can see clearly, right? So A blood group will have antigen A and antibody B. B blood group will have antigen B and antibody A. AB will have both the antigens and no antibody. O will have no antigens, both the antibodies. Okay, right? Now, who is the donor group? The donor group depends upon the antigen presence. Okay, see, for example, he is A blood group. Okay, I have A blood. Okay, so who have to donate to me? So A blood group means what type of antigens are present? It has A antigen. That's it, right? So the person who is donating to me should also have that antigen, but it should not have any extra antigen. Because see, when that blood enters into my body will recognize the antigens only, right? So when you're coming and someone guest is coming, he will bring a ID card, right? So if you see that ID card, okay, you will say, okay, he is from my college. If there is no ID card, you will say, go, you're not allowed into my college. So that ID card is the antigen which I have. So the person who can donate to me, the person who has A antigen. And if there is no antigen, that is also not a problem because there is nothing present on the surface. So I will not recognize it as a foreign particle. So if there is something new, then I will go and attack. There is nothing. So what I'll do, I'll not attack. So what happens? So who can donate to me? A can donate to me and O, because O doesn't have any antigens, na? So it's happily it can give to anybody. So you can see O is present in all the ones. So O is considered as the universal donor. And we have AB, which is universal recipient, because it has both the antigen. So the person who it is donating, it should have both the antigens, right? So all who have these two antigens can donate to them, right? So A, B, O, all can donate to AB, right? Fine. So let us end till here. For today's video, I'll be taking a second part of it where we introduce a next antigen, that is D antigen. Okay, so that becomes a little bit complicated when we go for the D antigen. Again, there comes a plus and a minus, which you all need to understand clear. Okay, so that part I will be teaching in the second video of this series. Okay, so I hope this video till here you have understood the concept well. There are some important points which you have to remember. So try to get it done. Okay.
If you think this video is useful for you, please like, share and subscribe to my channel. Don't forget to follow me on Instagram. Bye bye.